I'm Mariana. I'm a PhD student at Embrapa here in Brazil. And today I'm going to present my project for the doctorate. And it's about the editing of the stem cells with CRISPR-Cas systems. I think that we are we all need it because it would facilitate our lives much better. So I'll skip a little bit what are the stem cells because we all know them, of course, and I'll go directly in which genomes I'll be testing on this project. So, of course, the SIN1 and SIN1 that expresses the fluorescent red protein in cherry, and SIN3B and SIN3A that also have fluorescency on red. We all know that we still need transplantation for major genome editing, and that's something that's very hard to do, I imagine, since I never tried it, but when I read, it does feel like a lot of effort to do it. And our cells, just as the previous cells from it, lack some endogenous repair systems, which are the homologous directed repair and the non-homologous neighbor joining. So to overcome this, we propose a solution that is to combine CRISPR-Cas proteins with a recombinase system. So we are trying to use Cas9 and Cas12 that have some differences that I'll show later, and REC-A from the same family that are the Mycoplasma reprocolum. So our idea is to combine both of them so we can edit and insert mutations with it. So the major differences between Cas9 and Cas12 are, well, they both cleave DNA, double-stranded DNA, but Cas9 and Cas9 generates blunt ends, whereas the Cas12 has sticky ends, so this, well, might change a little bit our system. They have different protospaces adjacent motifs. Cas9 has NGG, and the Cas12 is more focused on thymine, which is good because our cells have a lot of thymidine, and as I said, the sticky end, double-strand break. They are very similar on size, and another difference is the guide RNA that is going to that takes the Cas protein to the site. While the Cas9 has a much bigger guide RNA because we have a tracker and we have CRISPR RNA, the Cas12 only has the CRISPR RNA, which is a smaller single guide RNA. So what we're going to do, we are going to transform our cells. I forgot to take the Cas9 plasmid, but it doesn't matter. We are transforming our cells and see if the Cas is being inserted in the genome by sequencing and see if it's somehow not enabling our cells to grow by seeing the color changing units assay. We're going to test if the protein is being expressed by Western blotting and also the translation through qPCR. And then we're going to transform as well with our guide RNA in a homology sequence so we can induce some changes in our genome. And we'll see through fluorescence microscopy, of course, for the M-cherry protein, and we'll analyze as well on flow cytometry. And of course, we're going to see if there are changes in our genes that are being mutated through qPCR. And these are the targets we had chosen. So the fluorescence protein, the casogamycin that we already have some ideas, and the literature shows us that they are working in this system. Since I'll talk about the GLPO later because we are also using another system, and some essential and non-essential genes, because we want to see if they are if it's working, because if we target an essential gene, the cell will die. So it probably is working. And some genes that we are not sure what they are, and they were chosen just because of the size to see if we have any limitation on the system. So these are the genes that we chose and their classification and what they do. So first we sent our DNA to be synthesized, but we had a problem with that. So I'm doing all by hand now through Gibson assembly. And I just received all my primers to do that. And the first construction was a Cas9 in tandem with REC-A with no type of control. So this probably will have some problems with our cells, but we don't know. We have to test this anyway. So to overcome that, our Cas9 and REC-A could be leading a lot of mutations throughout the whole genome. We are trying to control it through a integrase mediated by tetracycline. So the tetracycline is inducing the integrase, and the integrase will control our proteins. And once they are flipped because of the integrase mechanism, we have the Cas9 and REC-A being produced. So this is somehow trying to control the whole system and not making it toxic. So we're doing this with Cas9, and of course, I just talked about the integrase being mediated by the tetracycline, and we did the same thing for the Cas12. And with the same system, we just changed the Cas12 and Cas9. So this is the first thing that we are trying actually, and well, since we had a problem before, I never tried it yet. So I'll start right now to work on this. But on a side quest, we are trying to work out with the Cas9 system based on the Pascal's work that was already published. And it's also induced by tetracycline, but this one is not, the Cas9 does not cleave our genome. It just takes cytosine deaminase to the site and converts cytosine and guanine to thymidine and adenine. So we can try to put some, we can try to induce mutations on the bases. And so this is the original one. We have the checking primers for our Gibson assembly that we did again. And we're trying with the same gene. So we have the casogamycin, and this number reflects the site where the guide RNA was chosen. And cherry as well, that I don't know why I forgot to put it on. And the induction and the mutation we are trying to see is to induce a stop codon just right at the gene. I think it's all changed. Oh my God, I'm sorry. So the casogamycin is the same thing. We're trying to put a stop codon on it. And we already tested the cherry. I'm sorry for the whole thing is messed up here. And we saw that maybe our clones that are being induced are actually being, have a different expression on cherry. But we need further work on this, and I would really like to sequence to see what changes we had after the induction of our system. So the next steps we have is the Gibson is being translated through RT-qPCR, and also checking if we saw the specific stop codon mutations through probe-based qPCR. And the thing that's exciting is that I will continue this project of Cas9 and Cas12 in JCVI San Diego. That's I'm arriving in 2 weeks, and I'm very excited to be there, and I'm taking all these DNA there. So thank you, everyone, and thank you for listening to me. I'm sorry this was a bit messed and all the things, and to my advisors as well.
Very nice, Mariana, and I'm so looking forward to hosting you in our lab in 2 weeks. Questions from anyone? Mariana, so once you have a functioning editing system in minimal cell in Mycoplasma mycoides, what are your plans to do with this thing?
So I really would like to do a transplantation and have a strain on this, because I think it would help everyone. And I would like to disrupt the genes that we already don't know that are like somehow still missing, or the ones that we know they're semi-essential, to see if they are really semi-essential, and just play with it, really. I think it would help a lot on minimizing the DNA furthermore.
So when you come, would you like to install the Cas systems in the yeast clone of the minimal cell so that we can transplant, we can make a strain that just constitutively expresses the Cas proteins?
Yes, I think, well, yeah, not somehow controlled, maybe, because if it's was expressing, we should have problems.
Well, we can set up inducible promoters also.
Yes, yes, I think it would be very nice.